So you can say hi to the world now.
The millions of fans waiting.
Is the chairman already there? Bartolo?
It should be Bartolo, right?
I don't see him yet. And I see you now on YouTube also at the same time.
Yeah, me too.
So you can say hi to the world now.
Millions of fans waiting.
I would suggest, Nathaniel, that you put the YouTube channel on mute so you don't hear yourself.
Yeah, that's what I did. I think we were listening through Tom's computer.
Okay.
Yeah, sorry, I just turned it off.
That's okay. Don't worry.
I like the teddy bears.
Thank you. They're my emotional support animals.
But there's a huge delay. Okay.
Yeah, I put a 30 seconds delay for the latency, so I have like a good connection. Because if you do it's like without latency, then the quality is less good.
So also Sophia and Jan Kennis are wishing you good luck.
Oh, thanks, guys.
Practical question: is there any way to fail the public defense, or is virtually impossible?
That will depend on Benjamin, all right? On Nathaniel.
It seems like Bartolo has joined as attendee and not as panelist.
Okay, I can just add him, like promote him to.
Okay.
So promote to panelists. Voilà.
Awesome.
Voilà.
Bartolo is there, yes.
Hey, Lindert.
Hey, cool mic you've got.
Thank you.
Thank you very much. I burned my motherboard, so it has been a week.
Oof.
Very difficult week. So and I forgot to register. Or probably I did in my old.
Okay, so I will leave you to it so I can go for my practicals. Good luck, Nathaniel.
Thank you.
If there is any problem, don't hesitate to reach me. By Slack or something. I will keep an eye on it.
Charlotte, can you send me your cell phone number, maybe?
Yep, will do.
Then I can always send a message if something happens.
Perfect.
I was in a meeting with very bad internet connection. I hope it wasn't my problem.
So I guess when it's 4, Bartolo, you will start the thing, I guess. Or...
Yes.
Yeah.
Dominique Mas is there. Well, let's wait 4 o'clock, no?
Yes. We should wait time.
Everyone is there of
the jury, yes.
That everyone expected is there, yes. Okay, do you think we can start?
Sure.
Yeah.
Yes? Okay. Dear Madam, dear Mr., dear member of the jury, welcome to the public defense of the doctoral thesis of Mr. Nathaniel Mon Père, titled Statistical Biophysics of Hematopoiesis and Growing Cell Population. My name is Bortolo Mognetti from Université Libre de Bruxelles, or ULB, and they will chair the session. First of all, I want to acknowledge the member of the jury who have evaluated the thesis, in particular, Professor Gelen Lendert from Catholic Universiteit Leuven, Professor Mas Dominique from Vrije Universiteit Brussels, or ULB, and Professor Werner Benjamin from the Breast Cancer Institute, Queen Mary, London. This thesis has been supervised by Professor de Bulle Sophie from VEB, from Professor Lennart Tom from ULB. Notice that this is a joint collaboration between VEB and ULB. Now, the candidate already discussed his work and addressed the question of the jury during the private defense on November 17. Today, the candidate will give a pedagogical presentation of his doctoral work. The presentation should last approximately 50 minutes, and we will then have a final round of questions from the member of the jury and the public. I invite you all to prepare, so I invite you all to prepare questions during the presentation. Okay, now I think I would like to ask the candidate to deliver his talk. Mr. Mon Père, the floor is yours.
Thank you, Bortolo. Can you guys see my screen? Is it? Oh,
No.
wait, here. Share, there we go. So thank you, Bortolo. So yeah, my name is Nathaniel Mon Père, and I will be presenting my thesis titled Statistical Biophysics of Hematopoiesis and Growing Cell Populations, which was supervised by Professor Tom Lennart and Professor Sophie de Bulle. So a brief overview of the thesis. As you can tell by the title, it's really, there are two kind of separate topics. One is about hematopoiesis, and one is about growing cell populations. And this was kind of divided into four separate projects, one of which has been published already, and one of which is currently under review, and the other view we're currently still writing manuscripts for. And I'm going to focus in this talk mostly on these two projects because of time restrictions, and so I can kind of give a better explanation to the general public. So let's jump right into it, and we'll talk first a bit about what is hematopoiesis. So hematopoiesis is the factory for blood. It's the system that creates blood cells in the body. And daily there are around 300 billion blood cells that are produced. And if you would look into the blood and look at these cells, you would see various different phenotypes, different shapes, different sizes, different capabilities. And they all serve different functions. We have the red blood cells that carry oxygen. We have the lymphocytes and the white blood cells that provide the immune system, etc. But interestingly, they all share a common ancestry in the sense that they're all produced by stem cells, which are found in the bone marrow. And so how does this difference really occur? It's through a process called differentiation. So essentially, even though, as you probably know, all cells in your body have almost exactly the same DNA, they're not all the DNA is turned on at the same time. So you can have different, what they call expression patterns, and differences in expression patterns are what create differences in cell phenotype. And so this process of differentiation, we can imagine as a marble or a ball rolling down a hill. So it starts at the top, and once it reaches the bottom, it has a particular phenotype. And at first, it has to make certain choices that rolls down the hill, and then ends into one of these differentiated phenotypes. And so these balls at the top, we imagine them as the stem cells. They have the potential to become all of these other cells, whereas once the ball has rolled down the hill, it can no longer change. So if we were to look at a map of hematopoiesis, we would have these stem cells at the top, which are very rare, and they can differentiate along these different pathways and turn eventually through many divisions into these final mature cells, which serve particular functions in the blood. And as I mentioned, these cells are rare, and these are kind of what we're going to focus on in this talk.
Oops.
Our understanding of hematopoiesis is Did I just disconnect?
Yes, for a while.
For 5 seconds or so.
Oh, okay.
Should I suggest to switch off the video? Probably yes, is a good recommendation.
Yeah, let me try. I'll turn off my YouTube stream as well.
No.
There we go.
That's a pity.
Okay. So we're back though now, or?
Yes. I think
Okay.
it's probably
I think
was just the provider.
My
It
pointer
works now,
is lagging now.
but we don't see your slides.
Okay, let me see. Share screen. There we go. Okay. So as I was saying, we have this this map of hematopoiesis. And so in that sense, we qualitatively understand how hematopoiesis works, but there's a lot of quantitative aspects that we don't know, like basic fundamental quantities that we don't really have an answer to. How many stem cells are there? It kind of depends on who you ask. There are some estimates, but they tend to vary a lot. How active are they? So how often do they differentiate? How often do mutations occur? And those kind of things. And the reason we don't really have these answers is because it's hard to study hematopoiesis. It's happens in the bone marrow, and if you want to measure something in the bone marrow in a live human, that's obviously already quite hard. And furthermore, if we're looking at mutations or we're looking at expressed genes, we need sequencing techniques, and really very accurate sequencing techniques have only existed in the past few decades. So some general goals of what we're trying to do is, on the one hand, is construct models which we can use to apply to experimental data that allow us to come up with maybe values for these quantities. And in the broader sense, we could then, if we know these values, apply certain models to understand diseases better and come up with better ways to treat. So in this first part, we're going to focus, as I mentioned, on the stem cells, and we're going to talk about mutations and how they contain information on these dynamics. So when we think about mutations and fitness, we usually think about Darwinian evolution, right? There's some organism and it changes by selective advantages, right? Or sometimes, there can be selective disadvantages. But in reality, a lot of mutations, they don't confer necessarily a positive or a negative advantage to an organism. And we call these neutral mutations. And whether these mutations persist, whether they continue to exist, is really stochastic. It's random, dumb luck. It's up to a roll of the dice. And so why are we necessarily interested in mutations in stem cells? Well, these kind of mutations in our DNA, they occur throughout our entire life as cells divide. These are called somatic mutations. You don't pass them on to your offspring, but they accumulate the longer you live. And they occur during cell division, because cell division isn't perfect. And on average, recent estimates, we could say that per cell division, you get about 1.2 new mutations in each of these daughter cells. And it's important in stem cells because, as if you remember this little map we have here, if a mutation occurs in a stem cell, well, all of its offspring have this mutation as well. And so the small population of stem cells the mutations that occur there lead to many mutations in these huge populations of mature cells. And furthermore, these selective advantages we just talked about, well, it kind of depends on context. So you might have a mutation that doesn't really change anything in the stem cell level, where the cells are safely tucked away in the bone marrow. But once the cell becomes mature and is in the bloodstream, it might have an effect. And one example we'll briefly touch upon is this paroxysmal nocturnal hemoglobinuria, which is a disease that has that specific case. So firstly, one goal of this part right here is we want to look at the likelihood of a neutral mutant to expand within this stem cell pool. So we can ask ourselves, well, how does this happen? First, we think about what events would alter the size of a population of cells. And this is, of course, divisions. So we have what we call symmetric and asymmetric divisions. In the symmetric division, a cell divides, and both of its daughter cells, or the two new cells, are of the same type. So a stem cell, this HSC, can divide and we get two new stem cells. This is called self-renewal. Or a stem cell can divide and we get two differentiated cells. So these are no longer stem cells. Or the cell can divide and one of it is a stem cell and the other one is differentiated. And so the effect on the populations, of course, if we have a self-renewal, well, we've added a cell to the population. If we have a differentiation, we've removed a cell from the population. And if we have an asymmetric division, the population stays the same size. And of course, we can also add cell death. We're going to ignore that for the purposes of our model right here. And so the same thing counts for mutants as well. If we have a mutated population, if it's neutral, exactly the same thing, right? Self-renewal increases the population, differentiation decreases it, and asymmetric division keeps it the same size. So we can start constructing a stochastic model for this competition. If we imagine a population of normal cells and mutants, and then we would say, well, we're going to select one cell divides, and if these are neutral, they don't have a better probability of being chosen for division. So we randomly select one cell for division. But we have to remember we need to keep the stem cell population at fixed size, because we know that in humans, or at least in adult humans, the stem cell population doesn't actually grow or change size. And if we recall, the self-renewal increases the population by 1 and differentiation decreases it by 1. So what we say is we're going to take them both at the same time. So every time we perform a self-renewal, we're also going to perform a differentiation. And this is known as the Moran model. And with that model in mind, we can construct what is called a Markov chain. So essentially, we look at the size of a mutant clone at a particular point in time. So let's say this clone has M cells. If we then perform one of these events, we perform self-renewal and a differentiation, it can either grow in size, or it can decrease in size, or it can stay the same size. And there's a probability for each of these to occur. This is called a transition probability, so the transition from M to M plus 1. And it's quite easy to figure out, right? Because it's just the probability of the cell that self-renews, that cell was a mutant, is just the number of mutants divided by the total population size. And the cell that was chosen for differentiation is not a mutant, so that's just 1 minus that. And we can do the same thing for the other transition probabilities, and this allows us to construct what is called a master equation. And this is what we're really interested in, is this guy right here, which is the probability of this mutant having size M at a particular point in time. And the way we find this is, well, we look at the probabilities at the previous point in time, and for every state, and we say, well, the probability of the mutant in that state, K, moving to M, that's this. And we multiply that by the probability of the mutant being in K. And so we can sum over all of those states, and that gives us the probability in M at the next point in time. And so if we're interested in T plus 2, well, it's just the same thing, except this becomes T plus 1, and then T plus 3, then that becomes T plus 2, and so forth. So you can start at time 0 if you know the size of the mutant, and then just evolve this thing until future times. And we can look at this in continuous time as well. Then it becomes a differential equation. And then we get this division rate. So the number of divisions we expect to occur in a particular period of time. And this is one of those parameters we were really interested in the first place, if you recall. So a small application we can already do with this model is one of the projects I mentioned briefly, and it was published in a paper, is we can apply this to this disease, paroxysmal nocturnal hemoglobinuria, which is a rare disease, and it's an acquired disease. So it occurs later in life, and it causes destruction of blood cells in the bloodstream. And the cause of this disease is a specific mutation that occurs in a stem cell during the lifetime. But so, of course, the question is, if one mutation occurs in one stem cell, how does that go to 20%, which some disease threshold? If you have less than 20% or 10% or whatever, then you don't really have symptoms. So how does this mutant go from one stem cell to more than 20% of the stem cell pool? And of course, the first thing researchers thought was, well, there must be some selective advantage, but they never really found any. And so some people suggested that, oh, maybe it's just this neutral drift, right? It could just be neutral, but bad luck that you get this one mutant that grows to a large size. And we can test this with our model, because we can evolve this Markov chain to get the probability of that actually happening in an individual. And we can compare that with or use a distribution of the ages in a population, and then we can get something like this, which is if we ignore these two curves right here and just focus on the top one right here, this is the probability of a mutant having a specific size in anyone in the population. So the likelihood of someone in the population having a mutant of 40% is given by this value right here. So if we have some disease thresholds, anything larger than this is the disease. We just need to integrate or sum from 20 to 100%, and that gives us the total number of cases in the population. And doing that, we find that our model predicts about 2 individuals per 100,000, which is surprisingly close to what we see in clinical data. The one caveat is that we needed to use these fundamental parameters. Remember, we needed the number of stem cells, we needed the rate of these divisions. And as I mentioned previously, these are still somewhat debated. So in a sense, it's a valid criticism of this work to say, well, this is very parameter dependent. And that's why we're kind of, we really want to see how we can quantify these parameters better. So the next part is about subclonal dynamics in stem cells. So we have talked about one mutation growing to a particular size by pure randomness. But if we were to look into a bunch of cells, you would find thousands of mutations there, because, as I mentioned, there's about 1 or 2 mutations occurring every cell division. So let's imagine a cell, and it divides, and it gets 2 new mutations. Let's call them A and B. And every new mutation is almost certainly different from mutations that exist, because the genome is so large. The likelihood of one small mutation being exactly the same as one that occurred previously, if they're all random, is vanishingly small. So we can evolve this for a few more generations, and then we end up with this, like an observation of these 8 cells, and they all carry a bunch of mutations in them. And so these mutations, A, for example, that occurred here, we see them in all of these cells, and B that occurred here, we see it in all of these cells. And this is the kind of observation that we can make if we have good data. And so one thing I want to touch upon briefly is the fact that if we define something as a clone as the collection of cells which share a particular mutation. So for example, this mutation A is found in all of these cells. So all of these cells form the clone A. Then we can look at this observation in a slightly different light. Say, oh, A has the cells A, B, C, D, B has the cells E, F, G, H, and so forth. We can do that for every mutation that we defined. And so the preposition is really that this is what we call the clonality of the system, these different clones. And the preposition is that this contains information about the behavior, about these fundamental parameters we were talking about. And so the goal is, what can we learn if we measure some quantities of this system, if we study this clonality? And then, of course, if we do know that, can we actually feasibly extract that from an actual data set? So we're going to look at a particular data set made recently. And this is, for example, the lineage tree that they reconstructed. So this is essentially the same thing as this little tree that I showed here, this tree right here, but then in actual hematopoietic stem cells. And so we're going to see if we can apply that to that data set. So the game plan is we're going to look at two measurable quantities related to this clonality. The first is what we call the single cell mutational burden. And this is essentially the distribution of the number of mutations per single cell. It's a lot of words, so let's look at some pictures. That's maybe easier. We have our observation that we had earlier. Every cell contains a bunch of mutants. What we're going to do is we're going to count the number of mutants per cell. So cell A has 3 mutants, cell B has 4 mutants, and so forth. And then we're going to plot the histogram. So how many cells have just 1 mutant? Oh, that's just this one. So there's 1. How many cells have just 2 mutants? That's also just 1. How many have 3 mutants? That's 4, and so forth. So that's one quantity we're going to look at. And the other is the variant allele frequency spectrum. So variant is just kind of a synonym for mutation. So whenever I say variant, don't freak out. I just mean mutation. And so this is the distribution of the number of cells per mutation. And so you can see where this is going. We look at the other picture I introduced earlier, where we have mutations that contain cells. And per mutation, we just count how many cells it has. And once again, we plot the distribution. Yeah, and so typically when we talk about this variant allele frequency, instead of looking specifically at the number of cells that each variant has, we look at the fraction in the population. So the state of one cell becomes the frequency that this variant has in the total population. So let's see if we can predict what one of these quantities, this mutational burden, is going to look like using the model that we introduced earlier, this Moran model of divisions. So the total number of mutations that we find in a single cell upon observation, these right here, is really a sum. It's a sum over every division that ever occurred in that cell's past. And every element in that sum is just the number of mutations that occurred during that division. So if there were Y divisions, and in each of those divisions there were XI mutations, and so we need, these are of course random numbers, right? It's a stochastic system, so it's always going to be different for every cell, and so we take XI as Poisson distributed. I won't go into too much details about why, but the important part is that the expected value is of course this mutation rate. So the number of divisions that we expect to occur per daughter cell. And then the number of divisions, we also take Poisson distributed. And this expected value is now a combination of this symmetric division rate, the rate of these symmetric divisions that we introduced earlier, and the rate of these asymmetric divisions. And so this kind of thing is known, it's called a compound Poisson process. And we know how to get the variance and the mean from that. There's these guys right here. And so we already have these distributions, so we can do a little bit of arithmetic. And we end up with this thing. So we can solve this for mu, and we find that this parameter mu that we're interested in, we can find that just by measuring the data set. If we can just know the variance of this data set and the mean, then we get mu. And similarly, we get this quantity right here, which is some combination of the division rates and the time that has elapsed. So we can try to apply that to a data set, this data set I talked about earlier, and let's see what we get. So in this specific data set, they had 89 single stem cells from a 59-year-old patient, which means we know this T. And we get something like this. So the blue boxes are the burden distribution of the data set, the histogram. And I fitted this to the compound Poisson, which is this orange line right here. Unfortunately, of course, 89 is not a lot of cells, so it's not a large enough sample size to really make like a statistical estimate of how good the fit is. But we can see it works, for example, better than if we would to just take a random Poisson distribution and fit that to it. And so doing this this process I just described, we can figure out what mu is, and we get a mutation rate of 4.3 mutations per division, which is a bit higher than the 1.2 I mentioned earlier. But it is certainly the same order of magnitude. And we get for this total division rate, we get a 4.2. So that would be 4.2 total divisions per stem cell per year. This is per year. So they divide very slowly. So let's see if we can do the same thing for the VAF spectrum. So we're going to evolve a Markov chain similarly to what we did in the previous chapter. So we're going to use these transition probabilities, the likelihood of one mutant going from state k to k minus 1 or k to k plus 1. And so similarly, again, we have this this axis, and now instead of a single mutant being at k, this is actually the number of mutants that are at k. And the probability of one mutant transitioning to k plus 1 just actually becomes the fraction of these mutants that go from k to k plus 1. So actually, the dynamics of these states right here are exactly the same as we had in the previous chapter. However, now there are new mutants coming into the system with every division. So we have a source term right here for the very first state, for state 1. And so these combined differential equations, we can evolve these to get an expected VAF spectrum over time. So, all right, we can try to test that to our sample, except there's still one more problem. And that is that if we sample a population, a mutation can actually have a different frequency than if we were looking at the true population. I'll just give you a quick example of this. Let's say we have this population of stem cells. There are 20 stem cells and there are 5 mutants. So the frequency of this mutant is 0.25. So let's take a random sample. All right, we now have, in this sample, we have 5 cells and 2 mutants. The frequency of this mutant is now 0.4, which is higher and definitely not 0.25. So what we want to know is what's the probability of a variant with size n being sampled to size s. Turns out that is something called the hypergeometric distribution, which is given by this guy. And we can use that to construct the VAF spectrum, or the expected VAF spectrum at least, of our sample. And the way we do that is, well, we want to know how many variants in the sample have size s. And so every variant of size n has a probability of being sampled to size s. So if there are v variants with size n, we just multiply that with the probability, or the fraction of those that get sampled to s. And so we can just sum that over all of these n, and then we end up with the number of variants that got sampled to s. And so we can test this theory with some simulations. So the blue scratchy line here is a bunch of simulations where we just directly simulate these Moran dynamics I described earlier. And the full, or the dashed line, sorry, is this ODE that we evolved here. And so we can see we can match the simulations quite well. And if we sample, the system gets moved. So we see a higher VAF spectrum in the sample than we would see in the true population, and that's important. Because when we look at a data set, obviously we're not looking at all of the stem cells of this individual, right? We're looking at a sample. So now that we know that, we can try to apply this, try to fit this VAF, taking the sampling into account. And so the way to do that is we would do some Monte Carlo method, which means we run a bunch of these ODEs for different parameters, random combinations of parameters, until we find certain combinations that fit. And if we do that enough times, we get an idea of where it's supposed to be. And so we end up with something that looks like this. So we only have 2 free parameters here, P and N. N is the number of cells in the population, and P is this guy. So this is the rate of asymmetric divisions. And these are the symmetric divisions. So it's the fraction of total divisions that are asymmetric. And we see that we our expected VAF fits very nicely with the with a portion of the data. However, this portion doesn't actually fit nicely, and it's because I kind of ignored or didn't tell you about one part of this system, which is we've been ignoring the non-neutral mutations. So there might be mutations that do have a selective advantage, and we would expect them to be higher, so they wouldn't actually fit our neutral model. And then similarly, the system had to grow at a certain point in time, right? When you're a fetus, you have much less stem cells, so you early in your life, your stem cell pool actually is growing. And so these mutations that occur during growth, they would also have reached a higher frequency. But so this first set of VAFs, they actually fit quite well with what we expect. There's one more problem though, and is that this isn't actually the only correct solution. This isn't the only one that fit. And actually, we found that if we look at the solution of our Monte Carlo method, we have all of these parameter sets. So this is P and this is N that actually give us exactly the same line. That's not necessarily terrible. It means we can't actually know what N is, but it does give us a maximum limit for N, right? Because we see if this fraction goes to 0. So if it's purely symmetric divisions, that's when we end up with the highest population size, and that is about 35,000 stem cells. So I'm going to summarize this part real quick. We saw that the probability distribution of the size of a mutant clone, we can obtain that if we have a model of how the cells behave, and if we have some fundamental quantities. And we saw that the VAF spectrum and the single cell mutational burden, they contain information about these quantities. And so we can compare predictions of them to data sets and use that to fit the quantities from the data. And so we found for an adult stem cell pool, 4.2 divisions per year per cell, and 4.3 mutations per division per daughter cell. And finally, we also saw that sampling the population causes the VAF spectrum to shift upwards. So if we do compare the VAF with data, we need to take that into account. And fitting the predicted expected VAF gave us a maximum of 35,000 stem cells in a human. Okay, so that was the first kind of project I wanted to talk about. Now we'll talk about the second project. This is something completely different. It's about statistical mechanics of growing populations. And let's start with a bit of a motivating example to kind of situate what we're trying to, this type of systems we're trying to look at. And the story behind this is kind of that we were approached a couple of years ago by a group of researchers who were looking into cancer cell motility in metastasis, which is when a cancer spreads to other points in the body. And there's a process called epithelial to mesenchymal transition. And this changes a cell from like a normal cell type to like a faster cell type, gives it more motility. And it has been found that this may play a role in metastasis, and that certain cancer types, they may use this EMT to increase their likelihood of metastasizing. And so what these researchers had is they had a culturing experiment with all of these cells, and then some of them were these slow epithelial cells, and some of them had gone through this EMT, and they were these faster cells. And then they did a bunch of stuff. You know, they looked at the drug gradient, or they added a drug gradient onto this to see where the cells would die. And then they could measure where the cells' positions were and their velocities, et cetera. And they asked us, hey, can you do some modeling with this data? And we thought about it. And of course, if you want to learn something quantitative, you need to have some models. But we quickly realized that the fact that these cells are dividing constantly influences their motility. So here's a plot that kind of examples this. This is known as the mean squared displacement. I'll explain it a bit more in detail a bit later. So really what you only need to know now is that normally you would expect this to be linear if the cells were just random stochastically moving things. But instead we get sort of this curve, this sublinear behavior over time. And so we kind of posit that this is caused by the availability of space being reduced because the cells are growing. So the cells are dividing, and so the population size is growing, and this reduces the available space. And we couldn't really find any general models which say, oh, if you have growth in your population, this is what you do to model it. So we decided, well, maybe we can figure it out ourselves how this works. So I will give you a brief introduction to statistical mechanics, which is the idea that if you have a complicated system on a certain scale, if you zoom out to a larger scale, it can seem like it's random. So this may look like it's complete randomness, but if we would zoom in onto this Where is Waldo book, we would see that there's actually a lot of stuff going on, and none of it is actually random. And so statistical mechanics is just saying this, we're going to make a stochastic model of these macro scale dynamics, but we derive them using an understanding of the microscale dynamics. And so how does this relate specifically to motion? Well, imagine for a second that you are the 19th century botanist Robert Brown, and you are looking at a pollen grain that is moving in water. So these are pollen grains, and you see that they're all moving like randomly, a bit stochastically, and you don't really know what's going on. But almost 100 years later, some guy named Albert says, "I know what's going on. This pollen grain is being pushed by interactions with molecules in the water." And a bit later, some guy says, "Well, let's model this as this mathematical equation, this Langevin equation." And the idea is we look at the dynamics of the velocity. So the velocity obviously encodes the direction the cell is moving. And the proposition is that, well, this random movement is encoded by a friction and a stochastic force. And so the stochastic force, you can imagine it as every infinitesimal time step, there's like a random, it's drawn from a random distribution, it gets a random push in a different direction. And these two terms together work to cause this this stochastic motion, which models these collisions with water molecules. This is known as the fluctuation dissipation relation. So we might ask ourselves, well, these cells that we're looking at, can we consider them to be these Brownian particles? Well, cells are generally self-propelled, so they have their own internal motor. So it's not really collisions that's causing their motion. And they're also non-isotropic, which means that they have a particular preferred direction of motion. So also, it's possible to use Langevin equations, and it's done often, but usually they're a bit more complicated and they're not this form. However, what about if we just only care about the proliferation? So we imagine the proliferation just adds new collisions, right? The cell can't move because it's colliding with other cells. So maybe the collisions can be, or the growth can be encoded by this Langevin equation. And so the idea is to look at the simplest case scenario, which is we have ballistic particles, which just means that the particles move straight, but there's also growth. So we have particles that are moving in straight lines until they collide and then they change direction. And we say, well, for these ballistic particles that undergo these collisions, we're going to use exactly the same Langevin equation. And to see how this works is we look at something called the velocity autocorrelation, which is this guy right here. And it's essentially you take the velocity of the cell at a certain point in time and you multiply it by its velocity at a later point in time, tau. And then you average that over all particles, and you get something kind of like this thing right here. So it really encodes the correlation of the particle with itself. So how likely it is to still be going in the same direction. And if we calculate that from this Langevin equation, we get this guy, and in it we recognize that we can interpret this as the probability of the particle not undergoing a collision in a particular amount of time. So this gamma that encoded the friction, 1 over gamma becomes, in this interpretation, the time in between collisions. And this is known as the mean free time. And if there are any physicists here, they might think, oh yeah, of course, the mean free time also encodes something known as the mean free path, which is the average distance that a particle moves in between collisions. And the mean free path is related to the mean free time in this way. And then you might go, oh, yeah, we can also derive the mean free path in a different way. I won't do that here, but just believe me that this is the mean free path. And it depends, importantly, on the density of the particles. So how much particles there are per volume of space. So this allows us to construct a similar Langevin equation, except for these parameters, gamma and d, they depend on density. And we need to do a little bit of arithmetic, but then we can figure out the relationship if we use this mean free path as our basis. And so this allows us to plug into the density, we can plug a growth function. So we could say, oh, the system grows at this rate, and we can plug a function into n, and then we have a density dependent Langevin equation that depends on the growth. So for example, we could use logistic growth, which is typically found in culturing experiments with cells. So in order to test that our model actually makes sense, we compare it to simulations. So the idea is we simulate individually all of these cells, or all of these particles really, and all of the particles, they just move straight until they collide elastically, which means we're working with billiard ball rules. So all of the cells, they collide like billiard balls, and otherwise they just move straight. And we can add particles to the system according to some growth function, which represents the growth of our population. So we can then compare this. We compare the two, right? So first, if we look at the mean square displacement, this is that figure I was showing you earlier, which I'll explain a bit more now. The mean square displacement is essentially we look at the position of a particle, or how far it's moved away from its initial position. We square that, and then we average over all particles. So you can imagine if you just have particles moving straight all of the time, then this is just a quadratic function, right? It just goes as a square, and so the average would go as a square. But because these particles are moving randomly, right? They're changing direction. Typical stochastic motion, we expect this to be linear, is what I mentioned earlier as well. So essentially, what we see here, we see the simulations and the Langevin equation, they show this linear growth. And we did this for different densities of the system. And you see that if we increase the density, the mean square displacement, the slope at which the mean square displacement grows decreases, right? So essentially this just means the denser the system, the more particles there are, the less far a particle can get. This is essentially what we were positing, right? But so most importantly, what we were very interested in is what if we add this growth function, right? So we looked at different rates of growth. So this is the occupied surface density. So we start at about, I guess, 2% of the system occupied, and then we grow it till about 20% is occupied. And we do this for different rates. So this is a very fast growing population, a slower growing population, and even slower. And we get, for the mean square displacement, we get this this sublinear behavior that we saw earlier. Except, of course, we've said the population stops growing at a certain point. So during the growth phase, we get the sublinear displacement. But once the population is stabilized, again, we get this linear mean square displacement. So we can conclude that clearly proliferation plays an important role in this mobility of cells. And the crowding effect of proliferation, we can model it by using a Langevin equation and coupling it to a growth function for the density. And essentially how that works is we just take average interactions. We say, oh, interactions, they're just stochastic events, and put that into a model of stochastic movement. And then we see that the mean squared displacement appears to be subdiffusive or sublinear. And something I didn't really touch on yet, but as we mentioned, the cells themselves, they may have their own stochastic movement encoded in between collisions. And we can easily take that into account by just adding terms to our Langevin equation. So we have these collisional terms that we already derived, but we can add other terms according to whatever type of particle we're trying to model. Okay, so I will give you some general conclusions to this entire thesis. So what we did, to just remind you a bit, is we first looked at quantitative population dynamics in hematopoiesis. And we made predictions for the clonality and applied them to data from modern sequencing techniques to learn about the fundamental behavior of the system. We used an analysis of the VAF spectrum and the single cell mutational burden. And there are, of course, other ways, other quantities we could look at as well. And then we looked at motility in proliferative populations. We saw that it is affected by crowding or population growth. And we adapted a Langevin equation from Brownian motion, which we applied to model the effect. And that is it. So thank you everyone for watching. And also thank you, of course, to my promoters, Sophie and Tom. Thanks to Pierre de Bel for helping me a lot with this last project. George and David, who were big parts of the hematopoiesis work. Sylvie and Benjamin and Winnie and Marius, who were all helping with this this clonality project as well. And of course, thanks to Delavi, who provided me with the grant to perform this work. So, any questions?
Thank you very much for this very beautiful and clear and insightful presentation. So I will leave the floor to the expert from the Foreign Institute as usual. So we will start from Professor Werner Benjamin from Queen Mary.
Thanks, Bartolo, and thanks, Nathaniel, for this very nice presentation.
Thank you.
And also for the very nice thesis. It was a very interesting read. Maybe we start very general and start at the beginning. So you talked about Darwinian evolution, and you had this very nice pictures of the dinosaurs. Maybe for the audience is also an interesting question. What exactly do we mean by Darwinian evolution? So what is it?
Yeah, sure. So maybe I'll pull up this slide as well. If I can. There we go. So essentially, it's a bit of oh, screen sharing has stopped. Jesus. There we go.
Yes.
Yeah. So when we talk about Darwinian evolution, essentially it's kind of the term is a bit muddied nowadays for sure. But when Darwin talked about evolution, at least, he talked about these selective advantages, right? So a mutation, essentially, if it changes the individual and if it confers an advantage in a specific situation, then that becomes an advantage, and then that new population is more likely to exist. So, for example, this Pokemon is stronger than this Pokemon. So in a situation where strength is required, this Pokemon is more likely to survive. However, this Pokemon is less cute. So maybe in a situation where cuteness is more important, like Pokemon card collectors, this Pokemon will survive longer. And so similarly, we can also have negative mutations, right? Mutations that cause diseases. You can imagine that probably a lot of mutations, most mutations are negative even. And so that's what we think about when we think about Darwinian evolution. We think about competition between species, and we think about two things really: changes in environment that change the fitness of an individual or an organism, and then changes in the phenotype which adapt the fitness of that organism to the environment. Which is, of course, doesn't really take into account these neutral mutations, which provide a lot more variety.
And then the question, when we think about species, all the things, so you touched on that. Why do we introduce this concept of evolution into something that is related to the body, and why are we interested in what we call somatic evolution?
Yeah. So let me pull up this slide right here. So of course, somatic evolution is a bit different than or at least evolution amongst organisms, because when we think about selective advantages in organisms, we think about, you know, oh, this organism wins, but it's a bit different in human tissue, because your cells, they all serve a specific purpose in your body. And so if a cell gains a mutation that grants it an advantage, that actually means it's doing something detrimental to the other cells, and it's really not good for your body. So the idea of one particular cell gaining mutations and gaining advantages is actually cancer. So it's actually a bunch of cells that have kind of gained abilities that they shouldn't have in the context of your body. So the end goal of your body functioning as a whole is actually opposed to the idea of somatic evolution that occurs in your in your cells. So that's the main reason we're of course interested is cancer. It's a huge part of cancer is really somatic evolution. But as we showed in this thesis, we can also learn a lot about the body by studying somatic evolution. And so studying these neutral mutations gives us a lot of information about what's going on in the body. So in that way, it's also very useful.
And a very naive question. You said there are thousands of cells, thousands of mutations, closely enough. You said somatic evolution ultimately leads to cancer. Why is not everyone having cancer all the time?
Yeah, that's of course a great question, and this is because one mutation isn't necessarily enough to cause a cancer, and so cancer is really a combination of a bunch of mutations. It's known as the multi-hit hypothesis, I think, and it was actually discovered, interestingly, in before sequencing even really existed. It was discovered, I think, in the 40s or the 50s. By a mathematician who just looked at the incidence of cancer, and he saw that it required multiple stochastic events to occur. So really, in the simplest way of looking at it, you can imagine, oh, a cell gains one cancerous mutation that's associated with cancer. That doesn't make it a cancer yet. But if the cell grows and gains a sizable population size, and then other mutations start to arise that are also associated with cancer, from a certain point, one of these cells will have enough, or some of these cells will have enough mutations that make it a true malignant cancer.
And why are we interested in quantifying these type of things in the body? Like you said, number of stem cells, for example, or number of cell divisions, or mutation rates. Why does it matter to know exact numbers? Why isn't it enough to say, okay, there are stem cells and they do get mutations? Why do we care about these numbers? Does it matter if it's like 2 or 5 or?
Yeah, I think if it's 2 or 5, it doesn't matter. It depends. But of course, one example is this brief slide I showed you about paroxysmal nocturnal hemoglobinuria, right? Being able to do this work really required robust parameter values. So I think it's not, a lot of biology can be done, a lot of research can be done without knowing these quantities. That's certainly true. However, if we do know these quantities, we can apply mathematical techniques that otherwise might not just be robust enough.
I mean, I think that's certainly true. I mean, another thing, of course, obviously, is hypothesis testing, right? So, I mean, everyone can come up with an idea or an explanation why a certain disease or a certain pattern or a certain measurement behaves the way it behaves. But then how do we test that?
Yeah.
So, and then testing means we need some quantitative hypothesis that we then actually can compare to the data. So we need to be able to actually calculate what we expect in the absence of a certain effect, like neutral mutations, positive selection, and these type of things. So,
yeah, that's exactly true. Actually, yeah, I guess it's kind of the difference between philosophy in the 15th century and physics, right?
Yes, I think that's very true. And in that sense, from evolutionary perspective, does it make a difference if like the stem cell pool would be like a single stem cell or a billion stem cells? What's the difference in terms of evolutionary impact of mutations?
Yeah, I mean, the difference is huge, and I think that's another reason to understand this quantitatively is this gives us a better idea of what forces are working. So if it's very few cells, then these stochastic effects that we talked about, they become very important. The larger the population becomes, the less likely it is for a mutant to expand throughout these stochastic techniques. So the size of the stem cell pool is critical to how the body functions. I think the fact that we have these very few slow moving, slowly dividing stem cells, it probably forms as a barrier against cancer, because we have all these very highly populated mature cells. If these were constantly dividing and constantly acquiring mutations very fast, we might get cancer much faster because there are more mutations entering the system. Whereas because we have these very slowly dividing cells in the stem cell pool, we're less likely to get these cancerous mutation combinations.
So because you're a theoretician and I'm a theoretician, so for us it's very easy to propose additional experiments that people could do, and because we are not in the lab and actually have to do that, or in the clinic in the measurements. You discussed like single cell burden, VAF spectrum, so very specific type of measurements, right? One looks at single cells, the other one looks at cell populations. But in theory, I can dream up different types of measures too, and different types of dimensions I try to bring into the data. And one very interesting one, obviously, is time. So if I had time resolved data in a sense that I can follow up single people over time, what additional information would that add to the data, and what additional measurements could we take from it?
So I'm going to pull up an additional slide that I had that I didn't show it in the presentation, but I think this illustrates it. So I think I need to reshare my screen now. So one example, just right off the bat, I can think of that would be great with time measurements is for this VAF spectrum, right? Because now what we do is we fit one VAF to it, but really over time this VAF changes, right? And some of the parameters, such as like the division rate or the time that it elapsed, they serve the same function within our model. So they influence the dynamics in a certain way. And if we have time resolved data, so say we could measure someone's VAF at this point in time and we get this guy, and at another point in time we get this guy, this gives us a lot of leeway to fine tune our parameter values. For example, the fact that we came up with this, the fact that we couldn't exactly pinpoint the total population size is something we might be able to fix if we had more time resolved data, if we had more time points.
So what's the problem? Why don't we have time resolved data? What's the issue?
Well, of course, getting data in the first place is so. I think there's a number of problems really, and I think we're going to get time resolved data in the future. The first thing is these sequencing techniques. This data that I used, for example, it's very recent data. Let's see if I can pull up the data set. I shouldn't have done so many. Yeah, there we go. So this is 2018, and they used a very specific technique to get these this high resolution data. So we have all the information about the mutants, and it still wasn't even that many stem cells. But so the ability to do this kind of research is only a recent thing. So of course, from that perspective, we don't have data from someone from 10 years ago because I'm not sure that this kind of technology existed 10 years ago. So that's one reason. And the second reason is, of course, you need to find patients who are interested in doing this kind of research. I think their time resolved data there's some that exists for for cancer patients, I think that there might be, but in general, if we're interested in healthy tissue as we are now, for at least for this project, then we need an individual who has had their blood sequenced and had this done to a certain level, and then have that done again 10 years later. And so I think that's already a difficult process to get started.
Yeah, I think just because you touched on that, I think just maybe for the people interested in these type of things, pointing out how recent and fast developing these technologies are, like maybe people don't appreciate it, but these lines that you can see here in these phylogenetic trees, each of them single cell, and for each of them, the whole genome of that single cell was sequenced, which is the whole genome of a human. And this is like 120 or something like that. I don't remember exactly. Mm-hmm. Like I think the first human genome was sequenced in 2002, I think, and it costed $2 billion, involved 2,000 researchers, and it took over 10 years to do that. Yeah. And now we can send one PhD student to the machine. The machine sequences it within a week, and it comes back with the whole genome. Yeah. So that is really impressive. So that's just this technological changes and developments that make these type of things possible. And that's also why many of these things are still unknown, because before there was simply no way of looking at these type of things because the data didn't exist. So I think that is almost certainly right. If we look at this tree, because it's just here on the screen, it's also interesting, right? We see, if people look closely, you can see this very interesting patterns where you see a bit. So when you go to the top, where zero starts, zero means zero mutations. That is the very early beginning of the tree, right? And then you see these very small branches. And then later on, you see these very long branches. So this is a very peculiar pattern. Does this tell us something about what happened to this person? Or do we expect it to be universally true? Or is it just this person that looks like this?
So first of all, you could say, I think again, we have this phenomenon of the stem cell pool growing in size early on, right? So the thing is, if you say you have only the very first hematopoietic stem cell, if that divides, and then there's a new mutation of each of those cells, that mutation will be in every cell for all of the next generation. So really, these mutations occur very, very quickly in succession. So we would expect to see like the earliest mutations that are shared by a lot of cells, we would expect to see them very close to each other. And then the longer we wait, of course, once the stem cell population reaches a particular size, and it's still a relatively large size, we only have a sample of all these cells, right? So there are likely many more branching points and many more cells in between these. But of course, we don't have 35,000 stem cells that we sequence. We only have, I think, what was it, 89? So I think we would probably expect in a sample to see this kind of thing because we don't have all of the cells in between there to show us these mutational relationships.
So what exactly are these branching points? What exactly is it?
So this is essentially, if it represents really a cell division, right? So it represents a point where one cell became two cells, and we can distinguish that by these two carrying the same mutation and the cell before that not carrying the same mutation.
Yep, or a bunch of mutations, given how long
yeah,
how
exactly.
long how long some of these branches are. Yes, that's very interesting. And then we also can see, if we look at the tip of the tree, right? We see not all of them have the same length. So some of them are shorter, some of them longer. You explained before in your talk where it comes from, but maybe it's hard for people to make the connection. So what does this connect to what you talked before in your presentation?
So the why all of these tips are yes. at different points in time?
Yes.
Honestly,
so why they are in different lengths, right? You see this variation. Some of them are.
Yeah. So I'm not entirely sure. So these are these are the mutations that are found not in the stem cells, right? But in the or they are actually stem cells, bone marrow. So yeah, I'm not entirely sure.
I think you have a slide in your presentation that talked about it.
You'll have to enlighten me, I think.
Is the compound Poisson, right? Because this is.
Oh yeah, because molecular time is measured in number of mutations.
Yes. So
yeah, of course, yeah.
It's simply the burden.
Yeah.
And then
that connection, that's good.
And then this different length is simply the representation of the stochastic process, right? So you have these cells that what you said, they have two types of, at least, I mean, maybe there are more, at least two types of stochasticity, right? So there's this mutation accumulation, there's a random number with each division, and then there's this random number of divisions that then causes this variance. And basically, the weight of the distribution, exactly, the weight of the distribution that you discussed.
Yeah.
Exactly. I don't know, because I mean we have a very nice conversation. I don't know if there's some questions of the public or someone else that want to chip in. I don't know if how we do the otherwise I'm going on, but maybe there are more urgent questions from people that they want to ask.
I would do a single round though, so
okay.
I suggest that you continue until
sure.
You're satisfied.
So sorry, I saw Bartolo's screen. You said this becomes a bit technical, so I don't know if this is, but you said this mutation rate that so we have this estimate, right? If you look at this burden, and the way to do that was to look at the mean and the variance of this burden distribution, right? And that led to an estimate of the mutation rate, and also to this proliferation rate, which was symmetric, asymmetric. You said the estimate was slightly higher than we would have expected. Can you give some maybe somewhat intuitive explanations or possible explanations why this was the case?
Definitely. So, Andy, this is something we've been thinking about for a long time, and it seems like we might have kind of found why this is. And the problem is that, again, it's this initial growth phase of the stem cell pool. So when we make this compound Poisson, we're kind of silently assuming that the population is fixed, and that will give us this estimate. So this estimate would be true if the population started at a particular size in time, but it doesn't. It starts at one stem cell and then grows, and then it stays fixed for the rest of your life. And so we found that if we run simulations of this Moran model where we have a growth phase at first, we see that in these simulations, what we measure really is that this estimated mutation rate that we see goes up. So we kind of get like a linear growth, or just a growth in general of this measured mutation rate. So really the measured mutation rate we're seeing is actually overestimating what it actually is because of this growth phase. So if we want to fine tune this, we would need to kind of adjust this model to take into account a growth phase, and then hopefully we would be able to offset that issue.
Do you think this is more like a speculation? Because we always talked about the blood system, right? Do you think if we were to do the same inference or the same type of analysis in different human tissues, it would look entirely different, or we would find something that looks very similar?
Mm-hmm. Yeah, that's a great question, because there's a lot of data on different tissues as well, and what we've seen in some other tissues is that let's see if I can go to this VAF. So here's the VAF spectrum we're looking at. I don't really have a plot right now to show you that it's growth, but I think I'm going to pull out this plot right here. I'm not sharing my slides anymore, I guess, right?
No, at the moment I see you.
Okay, so.
No, indeed.
There we go.
It's no, yes.
So if we were to evolve some VAF for a certain amount of time, eventually we go to this equilibrium. So as you can see, all of these lines are VAFs that have been evolved for different amounts of time. And over time it converges and then it stops changing. And so interestingly, if we look at different tissues, we see that they've reached equilibrium. And it's not entirely surprising, because if we think about it, the stem cells are dividing very slowly. It's something we know, right? We saw four divisions per year. That's incredibly slowly. And so this VAF is growing very slowly over time. But other systems, for example, in the skin, we have these small patches of stem cells, and they perhaps need to divide much more rapidly. And so if we look at skin data, for example, or esophagus data was another data set we looked at, is we see that actually we've reached this equilibrium phase, which is from our perspective of like analyzing things, it kind of simplifies certain aspects of it, because there's some simpler, or at least we know the exact analytical expression for this function, right? So it's a bit easier to do, to kind of infer parameters from it. But on the other hand, we don't know, there's a bit more information in this dynamical regime. So, but yeah, it's very interesting to look at different tissues as well. It tells us a bit about, just from a general perspective, what's going on.
I mean, I 100% agree with you. I just fear that now we become so technical that with this type of explanation, we lost of our YouTube channel. So maybe we take a step back and we try to explain, maybe in very few words, what an equilibrium actually is, but maybe the YouTube channel knows that. But what is the nature of that change in time in the VAF distribution? So where does it come from, and why does it change in time, and why does it go to equilibrium?
This is a kind of the fun things to think about when you're doing math, is figuring out what's going on. So if we remember, the VAF is the number of mutations that have a particular size, right? So we can imagine mutations entering the system, and they enter at size whatever the smallest possible size is, so 1 over the population size, and then if either they disappear or they grow stochastically, right? And so if you wait, we get kind of a diffusion of this histogram. So the longer you wait, the more likely it is for at least some variants to have reached a particular size. So that's why this kind of these curves are expanding outwards, right? So it's essentially the probabilities of mutants reaching higher sizes. And so 1 means a mutant has reached the size of the total population. So essentially every cell now carries that mutant. And so once we reach the equilibrium, so you say, well, why does it stop if we keep accumulating mutations? Why would the VAF not change? Well, this is the point where, if you remember, we can't actually detect mutants at size 1, right? Because they're the same as the population. So really this stops right before 1. And so when we're in equilibrium, what we're actually talking about is an equilibrium between 0 and 1, because mutations are also disappearing and mutations are fixating, going to 1. So, and these actually keep growing, but this all of the states in between 0 and 1, they reach an equilibrium, which means that all of the new mutations that are coming in are exactly compensated by the mutations that are fixing, and all of the mutations that are mutations that are disappearing, and all of the mutations that that come in here are also being compensated by those that are fixating.
And in this context, for this type of VAF distribution, are we talking about healthy tissue, or are we talking about cancer?
This is purely healthy tissue, because this is, in fact, it depends on who you ask. This is the healthy tissue where there are no selective advantages in any mutants.
So how does the picture change if I were to talk about cancer?
Yeah. So if you talk about cancer, our model breaks down, right? We assume that in this Moran model that we're always randomly drawing cells for division. And that's of course no longer the case, because the cells with a selective advantage, such as in cancer, they have a higher likelihood of being drawn. And so it can be shown, you can show this analytically, is that eventually, instead of this 1 over F equilibrium, we get a 1 over F squared. So you have more, so it's much shallower curve, and so you have more mutations that are going to high frequency.
And the nature of this 1 over F squared comes from this assumption of like a growing population, right? So you have yes,
exponential growth.
Yes, absolutely. And do you know if we find this type of like, is this like a nice theoretical prediction, but has no like doesn't bear any comparison to reality? Do we actually find these type of things?
Yeah, I mean, I think you have a paper on this, right, Ben? Where you compare the 1/f² to some data.
Yeah, if you come to London, you have to read up because there are two fractions.
Yeah, that's
people
from
that
what I
believe
understand.
that and people that don't that, and then there's a, but that's more about interpretation. So the shape clearly you can see in healthy tissue, this 1/f seems to be true, and in cancerous tissue, this 1/f² seems to be true. And the discussion is not about that. I think the discussion that the community at the moment engages in, or since like 3 years engages in, is in that what does that actually mean? And what does that actually mean? Means are all these mutations neutral, or are there mutations with fitness advantages that we can't see? So then there's a lot of discussion on that. And if you come to London, you might enjoy some of them as well.
Yeah.
But you touched on how would we expect, so if there's a mutation with very strong positive selection in a healthy tissue, what would we expect to see if we were to look at above spectrum?
Yeah, so we would expect actually, I'll go back to sharing. And this is where it kind of gets a bit difficult because we would expect to see something similar to what we actually see. But it's hard to prove one way or the other which of the two hypotheses is correct. So let me share this. Damn it. So you see my screen now? Yeah. So essentially, we have these neutral mutations, right? If there were a mutation that had a selective advantage, it would follow this 1/f² rule, right? And the thing is, if there's one clone or one population of cells that have this selective advantage, all of the neutral mutations that arise in this population, they also have that same selective advantage because they're living together with the cell that is better, or with this mutation that has the advantage. So we would see these are called like hitchhiker mutations. So we would see a 1/f², which might actually fit this distribution. I haven't actually tried it. We would see a 1/f² that lives together with this 1/f, and it would carry a bunch of other mutations as well that are hitchhikers. The problem is we don't really know whether this is caused by selection or whether it's caused by these early mutations, because that's also a growing population. We would expect to see exactly the same thing, which is, again, as you touched upon before, if we have good parameter values, if we know what's actually going on quantitatively, then we can easily distinguish between the two.
So if you were, that's a mean question, but if you were to design an experiment and you have infinite amount of money and you're not a PhD student, so you're not in a time restriction that you have to come up with results within 3 years, but you can wait for 10 years or for 15 years. If you need to design an experiment to decide this question, what would be the, like, ideally, what would you do to resolve this question?
Removing any moral issues? Oh no, this is going wrong.
Talk about that. It's purely, we do this in chicken.
Okay, there we go. No, so as I mentioned, we suspect this might also be caused by growth, right? So if we would have time data points for different time during the early growth phase, we could see what we could quantify this growth of the VAF during ontogenic growth, right? So if we know exactly the quantities, if we could do the same experiment for like a 1-year-old child and a 5-year-old child and a 10-year-old child, and then we would have time resolved data, then we could probably, I think, get some quantitative ideas of what we'd expect to see if this was actually selection.
And assuming we have some morality, we can't do it in children, but we only have adult data, is there a chance we can design an experiment if we only have persons in their adulthood already and look at these things?
I guess you could compare like cancer patients with non-cancer patients, and you could see if this selection is much higher. But then again, it doesn't really tell you. They could have a selective advantage in their tissue even though they don't have cancer. So I don't know if I could think of something right off my head.
I would suggest now that we continue with the other member of the panel, because someone has to leave. So we'll continue with the expert from other Belgian university. In particular, I leave the floor to Gellens Lendert.
Thanks, Bortolo. First of all, Nathaniel, good to see you. Nice to see your presentation, and congratulations on the great work. It was a great read, and I enjoyed our discussions already in the private defense. So rather than having a discussion on more technical aspects, I've just got a few more general questions. So you presented also now like the two parts of your thesis, one about hematopoiesis and the other one about the motile cells and growing populations, and you developed all kinds of theory to sort of analyze those. So my first question is sort of what are sort of for you sort of the killer experiments that you would like to do or have someone do for you to test some of these theoretical predictions, and which of those predictions do you find most exciting, and would you like to see confirmed or not confirmed? Or
so I think this thing I was just talking about with Ben about clonality, I think those are quite exciting experiments and quite exciting things to look at, because of how the data is so recent, right? These techniques are only just coming into use and coming into practice, and so all of this data is kind of flooding into the world, and it's an exciting time to be able to apply these things. So I think these experiments that we talked about, these samples that have single cell data and all of the mutational information, those are really cool, and there's a lot of data coming out. Saw some preprints already that have like similar experiments but with more cells, different tissues. I think these are very interesting to study, and I think we can learn a lot from them, especially as the data becomes better, as we get time resolved data over the years. I think that's going to be very interesting. I think,
and so that's a type of data. Like, can you be like more specific? Like, is there something in your thesis that you say like, well, this figure, this finding? I mean, I think that's great, and using that type of data, which is not available now, we could test that, for instance.
So, yeah, I think, actually, there's definitely. I'm going to pull up actually a different figure. From a different presentation, from the private presentation, which kind of illustrates what I want to say. If I can find it. Actually, maybe I didn't show this figure. Okay, 1 second.
And if you don't find it, it's okay. You can explain
no, I
it in
got it
words
right
as
here.
well.
It's fine. I got it right here. So I just pulled this up from the thesis. And so if you recall, I guess I didn't talk about this, but so what we're looking at here is this is a comparison of these experiments, these Moran evolution experiments with the expected VAF spectrum that I showed. And what you can see here is that I also had a calculation for the variance on this thing, because really all I calculated was the expected VAF. And my calculation for the variance turned out to be terrible compared to what's going on in the in reality, just in the simulations itself. So I'm really excited to kind of figure out, one, how to accurately get a better prediction for this true variance in the simulations in a model, and then two, I think it's really interesting to be able to compare variances with variances you measure in data, because we're oftentimes, when you think about doing experiments, you think about comparing expected values to whatever you see, right? But there's a lot of information carried in variances of data sets, right? We saw that in the single cell mutational burden, where we got these parameter values by using the variance of that distribution. And so I think there's a lot of information that's going to be encoded in the variance of these data sets. So of course, then we need like larger data sets, more cells. And then if we have a robust estimate of these variances, then it's going to be very interesting to compare predictions with those.
And how about the second part? Like, is there something that you think about for the sort of the growing cell populations?
Yeah. So yeah, also quite excited to I at least have one idea. Let's see if I can stop. There we go. Of something I would like to do in the future is share the screen. It doesn't really matter. So the model that I showed now in this section is a spatially homogeneous model, right? So we assume that the system is populated with cells, and there's and the density changes, but the density changes across the entire system homogeneously, right? And of course, that's obviously not true, but in some situations it's true on average. But in some situations, it's maybe not true. And so I would really like to kind of extend this model to be local in space. So we can really look at fluctuations at specific points in space. And then we could see, when does this model work as an average approximation, and under what circumstances does this average approximation actually break down, and do we get like local patches that grow in density and then cause the system to shift in its dynamics?
Mm-hmm. Actually, this is more an answer to like a next question I had, which was like, how would you expand your theoretical models if you had the time to do so? But you answered that at least for the second part now, which I understand you want to have sort of this inhomogeneities that you introduce. So either if you have done that, or with the model that as you have it now. So the first question was more like, with your findings, like if you then go to, let's say, ask somebody to do some measurements, what would you look at, and what would you like to see to sort of confirm or not the sort of key findings that you had in your thesis?
So for this part, since our model is in the one sense the most unrealistic model possible, because it assumes all the particles are moving straight until they interact. But the cool thing about it is that we can add any type of movement in between these interactions. And so I think the experiment that I showed you with this drug gradient is a bit too complicated to really confirm this model, because you have more things going on. You have this drug gradient, you have these two types of cells, you even have different sizes of cells. So a simpler experiment with maybe cells is a good place to start, though, I think. Some culturing experiment with cells, which would allow us to, on the one hand, we'd have this model for accounting for the collisions. Then we'd have to study the cells individually and come up with a stochastic model for their own movement. But there are some models that exist for that specifically. And then we would combine those two models as summing them in the Langevin equation, as I showed. And then it would be cool to see if we can, if we can combine these models and then fit it to what we would expect for the movements. And yeah, that would be cool if those match. But of course, it's a lot. It would be a large effort to do, of course, and you would need the right type of data.
Okay, well, thanks. At various places in your thesis, you also use sort of negative feedback, positive feedback that you introduce in different places. Could you maybe comment on sort of some general properties that sort of these types of feedback typically give in a wide variety of biological systems? And how does that compare to sort of what it does in your systems where you studied it? Is that consistent, or are there things that you could learn from the general theory?
Yeah. So I think, I mean, there's a lot of general theory on feedback, of course. So maybe just like the simplest things that you could get are you can get like oscillatory behavior. You can get oscillations if you have positive feedback or negative feedback, or bistability, for example. I think in the systems that I looked at, because and I guess you're also referring to the third project that I didn't really talk about in this presentation, but so the system that we're looking about is being a system in the human body. It's already highly evolved to like present as little possible as any of those like extreme situations. So if we look at just the process of cell differentiation, and maybe you lose some cells in the mature compartment, and so the earlier compartments need to compensate that. Well, obviously there's some feedback loop, right? The loss of these cells transmits some signals, and then the bone marrow knows, oh, we need to produce more or whatever. But if in the simple model, for example, that I showed, you can get these crazy oscillations and stuff like that, I think we can say that our body in general has sort of a lot of mechanisms, additional feedback mechanisms that will sort of work to prevent that. So I think in the simplest case, you get simple solutions like oscillations or these losses. I think those are generally compensated in the real system by additional feedback mechanisms. But again, those can become complicated as well. And then I think once you have all of these networks interacting, you get things like bistability and switches and those kind of behaviors. Okay.
All right, thank you. I've got some more specific questions, but I think I'll leave the floor to the next jury member to ask questions. Thanks a lot.
Thanks.
Thank you, Slender. It's now time of Dominique Mas.
Thank you, Mr. Chairman. So Nathaniel, congratulations with your thesis. It's very nice. And I particularly liked how you combined two fields of research together, the physics and the more molecular biology. And so I have two quite general questions, and they are a bit along the same lines as the one of colleague Gelens. So when you do your predictions, I mean, you need some experimental data, but what do you think is really the major drawback? The lack of experimental data or the complexity of the system?
I think, yeah, I think I understand what you mean. I think in the case of like specifically applying these physical models to biological systems, right? I think the complexity of the system is actually the real problem, the real reason it's difficult. I think maybe like 100 years ago or maybe 50 years ago, there was this sort of this idea that, oh, once we start applying physics to biology, we're going to understand biology to the same level that we understand physics. But I think that's sort of inherently untrue, because these systems are so complicated and our maths just isn't really equipped to understand that. I think one of the problems is that these systems are so complex on this micro scale level that there are so many possible realizations that the system can enter once you zoom out to the macro scale level, that the only thing we can do if we want to describe it on the macro scale level is this statistical mechanics or statistical physics, right? Where we say there's a lot of randomness going on, and we just take these functionalities on the micro scale level and then plug them into some stochastic processes. And so I think one of the difficulties is the fact that we're working with these stochastic processes. We can't say exactly what's going to happen. We can't say when this pandemic is going to be over, because it's the stochastic system, not inherently, but because it's so complicated that on the macro scale, that's all we can do to look at it.
Yeah, and this is in line with my next question, because I mean, molecular biologists, if you need more data, and like you said, experimental data would improve my model, how will you convince them to give you new experimental data or to set up experiments, because they need some benefit from it? How will you do this?
Yeah, I mean, that's a very good question, and I wish someone would tell me the answer to that in general. I think it's important to be able to make a strong case, of course, And to do that, I think you need to make your models interpretable. So it needs to be interpretable by people who maybe don't necessarily know exactly what it is you're doing. But then again, I think it's hard because it's also easy if someone doesn't know exactly what you're doing, it's maybe sort of easy to lie about what you're doing and lie about what your results are going to be, and they might not know. And so maybe some people kind of, I don't know, I think the competition for getting people to do these results is difficult for that reason. You may believe what you're doing is going to work, but a lot of other people with different models may say the same thing. And so I honestly wouldn't know how to sell a model in the perfect way to get this.
I think if you have to sell your model, you have to convince them with the outcome, with your product, and would you be able to do so? To say, well, this is this is my problem, my product, and you will benefit from it.
Yeah, I think if you have a good problem statement and you can show exactly what the outcome is, yeah, definitely. I think, for example, in the case of, for example, these VAF distributions, if we can come up with values that really fit what has been already discovered, then they'll believe that your model works and we'll say, oh, okay, we'll take your model.
And then another question, in fact, by doing all this, what did you learn which is positive for the treatment of diseases? Or didn't you learn anything that you say, well, this might be interesting for, well, tackling some diseases?
Mm-hmm. Yeah, so I think while sort of like my PhD kind of started with the idea of being more close to actually the diseases, due to sort of circumstances and also kind of my interests, I kind of always moved back more to the fundamental stuff, which makes it maybe a bit less relevant directly to treating diseases. I'll definitely admit that. But I think, for example, in this part about feedback, we certainly, I think at least on a qualitative level, learned what we should expect when we treat people, in the sense that if you have certain diseases that cause variations in cell numbers in the blood, you should expect these feedback mechanisms to go into action. You should expect more differentiation, or more divisions as well. So this increases the risks of more diseases occurring as well. You can see, for example, in PNH that telomere lengths in an individual with PNH, telomere lengths in the bone marrow of the cells, they're shortened, meaning that these cells have probably been dividing more. So this kind of reduces their lifespan. So I think it's sort of, in this sense, it's sort of like on an intuitive level, to understand the dynamics that are going on if you would start applying treatment is important.
Mm-hmm. Yeah, thank you. That was all.
Thanks.
So before passing on with the advisor, I would remember that the attendee can post their question in the chat of the meeting. Okay. Now I will leave. Maybe Sophie want to say something? Ask something?
Yes, so congratulations, Nathaniel, for your PhD and for your presentation. It was very nice. I think people exhausted more or less my question. I was also interested, as Dominique just asked, how could you help with disease with your model? Mm-hmm. But you already explained that, so yeah, I will pass the word to Tom. And congratulations.
Thanks.
So thank you, Sophie. And also from my side, congratulations, Nathaniel. It has been 4 very interesting years with many turns and new ways and stuff that you were doing. Like I said, with the private events, I had 4 years time to ask questions to you. I learned a lot from you. And I think this is also very interesting because you have become the master of the things that you've been working on. So I think it's quite good, but I will leave the praise for after the discussion with the other jury members.
Thanks.
Okay. On my side, I had a very small question, probably, to be fast. You mentioned that stem cell, I mean, in your moral model, stem cell do not die.
Mm-hmm.
Was that biologically motivated, as I guess, or it's rather a simplification of the model?
It's certainly a simplification. I think so we know that stem cells die or at least become senescent, right? So there is a state in which stem cells lose their ability to continue to divide, and then they effectively die in the point of our model. So it's true we could include a death rate to the model, and it's probably even a good idea. We left it out for two reasons. For one, it kind of has a similar effect to symmetric differentiation, right? It just in the sense of our model, it just removes the cell from the population. So if we were to then evolve everything as we did, really when we fit something to the data, we just have a symmetric differentiation rate that is combined with the death rate. So that's one reason. And so in that sense, it's true, we could just add this death rate to the model. But then we have two parameters that we can't separate unless we have some way to measure this death rate specifically. So we end up with the same thing. We just always come up with this combined parameter.
Okay. I see. Okay, I want also to congratulate on this very beautiful work and very beautiful presentation.
Thanks.
I learned a lot.
Thank you.
I will give the possibility to Benjamin to revive the discussion. Probably you want to wrap up.
So from my side, I mean, I'm super happy. You gave me a lot of time to ask questions. My only thing would be if there are questions from the public, like some other from the audience, if they want to ask something.
Yeah, I just see that Stefan asked a question here in the chat.
Yes. So the question is the following from Stefan. I cannot see the Stefan Vette. In the compound Poisson formula, the mean had the same unit as the variance. How is that possible?
Thanks. So let me pull up the slide right here so everyone can kind of follow what he's saying. This is the wrong presentation. There we go. Here. So yeah, in the compound Poisson, the mean has the same unit as the variance. How is that possible? So let me share screen. So yeah, right here, for example, we have variance minus mean. If you think about what the mean is, right, is you take the variable, you take a realization of your random variable, and then you sum over a bunch of realizations and you divide by the number of realizations you had. So the mean should have the units of the random variable itself. Whereas the variance is you take basically the square of that minus the mean. So the variance should have the units of the square of the random variable. And yet here we have these objects where we're dividing, we're subtracting these. And from a standpoint of dimensional analysis, it looks a bit like heresy. I will agree with that completely. So the point is that it's not entirely applicable to look at it this way, or maybe to be more specific, I kind of I write these in a bit of a heretical way if you wanted to think about it in terms of dimensional analysis. For example, if you take the Poisson distribution itself, the Poisson distribution has one parameter. Let's say this Poisson distribution has a parameter mu. The mean of the Poisson distribution is mu, and the variance of the Poisson distribution is mu. And the random variable of the Poisson distribution is the number of times something has occurred, right? The number of mutations that occurred. And so we already say when we just describe this distribution, we say the number of times something occurred is mu, and mu itself is actually even just the rate of occurrences. So when we talk about distributions and we talk about their averages and their variances, and we assign a value to them, what we're really saying is the variance itself has the units of squared of that parameter that's true, but the variance is equal to this mutation rate. And this mutation rate, so really you have to think of it as a dimensionless analysis, right? So the value of the variance is equal to the value of this mutation rate. And so from that perspective, you just get the value of the variance minus the value of the mean divided by the value of the mean. Yeah, and that's how we get to these results. I hope that answered the question.
All right.
Yes.
Yeah. Are there any other questions?
So maybe if someone checks on YouTube, I closed my YouTube tab.
Oh,
I checked on YouTube. There's no questions on YouTube.
Okay.
They're just saying, well done, showing clapping hands and muscles.
Thanks.
Then I would suggest that the jury should deliberate, and we will be back probably in the same in this same virtual room, I guess.
Yeah. Yes.
After
So
the deliberation.
I guess, Anne, you can readmit us, I guess, or I don't know if you are readmitted automatically, but we will go to another Zoom meeting room now,
Yes.
and we will come back.
Everyone.
Yeah.
Okay.
Like the jury.
Come back.
Yeah, we come back.
So, but we have to quit this.
Yes.
Room.
Yes.
Hello.
Hey.
Congratulations. It was
Thank
a really
you.
nice presentation.
Thanks.
How do you feel?
I'm pretty good. I'm hungry.
How long does this usually take?
Like 10, 15 minutes usually.
Okay.
I think so. I'm not sure.
This is interesting because like 24 people are watching you just idly waiting.
I mean, allegedly, right? They could just have the YouTube channel open, but I also like to think that they're just watching me do nothing.
You're just watching you pick your nose, kind of. Oh. Is anybody watching? If yes, please say something.
Say something in the let me know in the comments, guys. What do you think? Smash that like button. Hit subscribe. Hit the little notification bell. You know how it works.
Oh, seven people have clicked like so far.
Wow. Super popular.
Yeah. So what's the next step after a PhD?
I do a postdoc, which is the same thing, except I don't have to defend, I guess.
What is the motivation for doing a postdoc?
I guess if you like doing research, it's sort of if you want to. So yeah, I guess like if you want to stay in science, it's kind of the next step. So it's just a researching position. They just call it postdoc because you need a doctorate to be allowed to do it.
Sorry. Well, what about like what are the other options? Starting a company?
Yeah, I guess go into like the real world and get a real job, which is something I have no ambition for. So
yeah, I understand that. Okay, 22 people watching now. Okay, our discussion about your future is very unpopular. However, you have increased 1 like.
Nice. And it's also just a monologue because like,
oh, can they not hear me?
Oh, they can't see you, so it looks like I'm talking to a.
They see like my name kind of like. Maybe
You're
on my
an
camera.
AI that is designed to keep me company while I wait for my result.
Okay, I'm gonna. This is your defense. This was just so that the public would understand.
Ooh.
You look really tired.
Yeah, that was very tiring. So believe it or not.
No, I can understand that. It's like your brain is like, oh, all of the knowledge that I have learned. Now I will channel into the world.
Now I'm just pressing the delete key in my brain, really, just removing everything I don't need anymore.
Computer will self-combust in 5, 4.
If someone asks me a question right now, it's just, sorry, deleted.
It was nice that Benjamin kind of asked you so many questions.
Yeah. It's kind of funny because he's the guy I'm going to work for in the postdoc.
Oh, nice.
Yeah, him and his wife actually. It's
cool.
It's really fun. It's a super fun research group, and so I actually see him every Wednesday, and then we talk about the research. So.
I love how you just like stopped.
Yeah, that's the end of my story. So yeah.
So there's Ruben that's rooting for you as Nathaniel for Scientist 2020.
Sweet, that's awesome.
And then Felix is saying we're watching your every move.
That's creepy.
And then Jan concurred.
Could you reply to Felix with the police?
The police?
Yeah.
Just say the police.
Yeah, that's fine. Or is it Sting, that song? No, it's the police, right?
Oh, I get it. Sorry, my song culture is pretty bad.
That's cool.
How do you? Well, he heard it, so.
Yeah, I guess that's true. I guess this isn't awkward at all. It's a normal post-PhD thing.
I mean, it's going to be normalized.
Yeah.
I wonder what people in the future will do during this interval.
They'll probably find something because this is not sustainable, I think. Or maybe this becomes a new sort of trial you have to go through.
Just wait for your future to be decided.
Mm-hmm.
You're probably almost sure to get it, but you still have to wait.
You have to wait in silence in a room and then everyone watches you.
Yeah. I get it now. It's very grilling. Maybe there would be like a sort of a folkloric ritual where everybody on your channel just like bombards you with memes.
Yeah.
That will keep your mind sort of occupied for 10 minutes.
Are the people on YouTube hearing this? They should bombard me with memes.
Guys, bombard Nathaniel with memes, please.
Can you post the link in the chat for the YouTube channel? I'm
yeah.
too lazy to find it.
It's beneath the video. It's right beneath it.
What? Huh. Thanks.
Maybe the internet is creepy. I mean, you know, a lot of people are just becoming Twitchers, like they're streaming their stuff online. This is basically you streaming.
Yeah, maybe I should just start watching YouTube videos and then comment about them.
Maybe we can't send gifts.
Huh.
Well, get creative, guys. Come on, like draw gifts with your keyboard.
Yeah, I thought you guys were millennials. You know, ASCII script.
Make a donut with just hyphens. Maybe that was a bit rude. We still love you. We stream every Wednesday.
Yeah, I can imagine a lot of people coming back Wednesday to watch this again.
Actually, it would be a really cool thing if you... Oh! Stefan sent you a cat in a box. How did you do that, Stefan? That's not even a meme. I mean, that's not even an emoji.
He's a doctor already, so he probably figured that out.
Oh my god. You're so cute. No, but it would be really fun to have... You can do later impressions of your own...
Do what?
You can do later impressions of your own PhD presentation. So
no.
I'm guessing like, oh, in the middle, I was really nervous. And then later, I just felt this overwhelming sense of flow. I felt connected to the universe.
So I'll just do like I will rewatch. I'll do a commentary about my PhD. So I'm going to rewatch my PhD and then I'm going to give comments about that. So I'll be like, oh yeah, this was really great. I talked about that very well. I'm very smart. Do you know there's what was his name? I think R. Kelly did a video about that where he has like a song and then he sits in a cinema and he talks about how good the song is.
Like a sort of reaction video to your own work.
Yeah, exactly. That's the future.
I think it would be really fun to have a
Yadi saw that too. He's in the comments.
No, I think they're talking about the teddy bear that moved.
Pity.
Imagine if it became like a horror movie, like PhD student gets slaughtered by his own like possessed teddy bear during online streamed PhD presentation.
That's a pretty good horror movie actually. Yeah.
Are you guys into horror movies? Comment below. No, but like serious talk, it would be so fun to have a PhD like react to online YouTube videos teaching people what AI is. You know?
I fucking hate those React videos.
You do?
Yeah, I do not get the point.
I haven't watched many, so I can't really comment. But
Yeah.
it does seem pretty slow and like just repetitive. It's just people like laughing.
It's also, I think the idea is like, I don't know what my opinion should be about this thing, so I'll watch someone talk about his opinion.
Mm-hmm. Oh, they're coming back. Hey guys.
That's-
Hey.
Hey Ben.
Hey. I think the others should. Yes.
We change room without changing room.
We lost them.
So everyone's there? So
No, we're waiting for Lennart, I think.
Lindert had to go, so
Oh, okay.
but he, yeah, but he congratulates you very much.
So, dear Mr. Mon Père, dear Nathaniel, the jury deliberated on your public defense. The outcome was positive, and the jury awarded you the title of Doctor in Science. Congratulations.
Thank you. Thanks.
Congratulations.
I don't see anything on YouTube. Anne, is this normal?
Sorry?
Or is it delayed? Delayed.
It's delayed, yeah.
So this was on the ULB side. Now I will leave the DEB.
So, Nathaniel, I will do it very officially. Whereas Mr. Nathaniel Vincent Mon Père, holder of the degree of Master in Physics, awarded by the Universiteit Antwerpen in 2016, has submitted an original doctoral thesis entitled Statistical Biophysics of Hematopoiesis and Growing Cell Population, whereas the thesis was defended by the candidate in a public session on November 27, 2020 at, well, online, I must say, whereas all the relevant rules and regulations have been observed, we, the chair, the secretary, and the members of the PhD jury appointed by the Faculty of Science and Bioengineering Science of the Vrije Universiteit Brussel declare that Mr. Nathaniel Vincent Mon Père has earned the degree of Doctor of Sciences. Congratulations.
Thank you.
So, Sophie, the floor is yours.
Yeah, so, Nathaniel, congratulations on your PhD. It's really a beautiful piece of work. It's not only scientifically sound, but it's also pedagogically very valuable, I think, for newcomers in the field. So it was really a pleasure to be your co-supervisor. You are wise, curious, knowledgeable, and creative. So many qualities that makes you a great researcher. I really enjoyed a lot working on cell motility with you. And I feel like it's opening up many follow-up projects, and hopefully we can explore some of them together in the future. So I'm also very glad that you got this postdoc with Benjamin, and I wish you to keep loving science and to enjoy London.
Thank you. Thanks.
Leave the word to.
So now we're for the more funny side of the praise for your thesis. Of course, from my side also congratulations. And again, I think it was immensely interesting, and you had made a great job. And the future is only bright for you. But I'm going to try to share my screen. Wait 1 second. So let's see if this works. Keynote Nathaniel, that's the one.
Can we cut the YouTube stream real quick?
No.
So Nathaniel, I am gonna try to project this thing. So you should now see my slides, but I don't see you anymore. So I hope everybody can see it in little words. The start.
Yep.
Yeah, it's normally we give a speech, but now I was thinking now on this kind of online stuff, it's a bit boring because nobody can see us doing gestures very well. And so I preferred maybe to show you some slides. And I was very, very disappointed because I lost your picture with your lab coat. This was really, you did something wrong today. You should have worn your lab coat of Televi while you were presenting, your thesis, because people don't know this. When you get a Televi grant at the start of the Televi project you're working on, every person gets this nice white lab coat with a red heart on it. So I hope you still have it somewhere.
Yeah,
Nathaniel.
definitely. I have two even.
So this is something I remember. I had a picture, but I cannot find it. You see yourself in the back there, I think. If I'm not mistaken, is where you pick up your coat. But I also had a picture of you wearing it, but I lost it somehow. So
I like this Photoshop more, actually, Tom. So
so this Photoshop actually comes from our first trip together when we went actually to work on your project on metastasis to Paris, to Pierre-Marie Curie Institute in Paris, together with Sylvie and another colleague from the IB Square. And we had great times there. I'm just showing you the official pictures, not the pictures of the evening in the bar where we had some great discussions about life and everything with a French guy over there. So I'm not going to come back to that. But we had some great times, and I think it was really the start of a very interesting 4 years, and I was very happy to have you on board. And you will also see people on the YouTube and in the audience that the facial hair of Nathaniel changes over time. So one here's still in his beard period, and you see also here that Nathaniel was very fluent in presenting his research, impressing people with the knowledge of his physics and mathematics, and showing formulas to biologists, which was not always an easy job to do. But then he changed to the mustache period. I think it was one of the IB Square events that we actually organized. And also here he gave this interesting talk about the work he did with David Dingley and George Pacheco on PNAH and the hematopoietic system, on which later on he developed also this feedback work that he was doing for a while with George in Lisbon, actually. So you spent a nice period there. I don't have any pictures of that because I was not there, but I guess, and I'm pretty sure that it has been great to visit his lab and to discuss this stuff with him. But of course, Nathaniel, you're also very much a social guy. And so one of the social things you like to do is to drink and eat. And so this is also at one of our events at IB Square, where we have the Christmas parties and we have the barbecue in the summer before the end of the academic year, which is always great fun. And I guess you also enjoyed that a lot there. But also at meetings. So here we have one picture when we met Benjamin and Winnie. I think it was the one in Leiden, right, in the Netherlands,
yeah,
where we actually encountered this lady also. I think wasn't she involved in this story with the microchip with the moving cells, where we actually start discussing with them about this work. But you see there, it was a great time to be together. And then I also met Nathaniel once at a wedding. I will not show too nasty pictures, Nathaniel. I know everybody who was there knows what happens during the night and the next day, so I'm not going to go in that. But we had a great time, and as usual, Nathaniel came very suave in his costume with his nice black glasses at the wedding of Elena, which we all enjoyed a lot, and it was a great time to actually do this with all the people of the group going to that wedding. But one of the things I will miss the most, and I hope this history, this tradition will continue, is the music band, the IB Square music band. And this is something you started together with those 4 people down there. One of them already disappeared. One of them is gone, right? It's only Rudy who has left us. I think the 2 other ones are still there. And we had a very nice time there. And it actually was maintained. The band grew a bit. So Sylvie and Sophia also joined at some point, which was very cute and very nice. And I think it was a great thing. And now I'm going to try to show a small video. So I hope the sound will go through. If not, then I will simply stop.
Tom, when you share the slides, you can actually choose to share the video, the computer sound. So I don't know.
So wait, so where do I do that?
So when you
where's my mouse?
So you can actually unshare, I would say. And when you have to choose in the share window, below, like really at the bottom of the window, you have like somewhere where you can check and put like share videos, computer sounds. I don't know if you see it or not.
I don't really see it. Wait. So
you have to really. So you have to really
stop sharing completely.
Stop sharing. Yes.
Okay, and then share again. So sorry about the technical issues. I'm not used to sharing videos. Share computer sound. Okay, here it is.
Yes, exactly.
And that's for a professor in computer science, right?
That's why you have PhD students, right?
So here we go. So I will stop here.
But you played.
And just to summarize, I hope you heard the sound.
Sorry?
He plays everything.
Oh, so it's you have a choice in multiple careers, not only.
Yeah.
So just for me to conclude here, if anybody wants to have the video, let me know and I will send it. It's something that I kept, and I think it's and I hope we can continue this tradition with new musicians inside inside IB Square. But I hope you will continue that tradition in the lab of Benjamin and Winnie. So I want to see Benjamin playing the bass, for instance, and Winnie playing the flute. That would be very nice. And this is where I will stop. And thanks again for a great 4 years. And I hope you enjoyed your stay in Brussels. And I really wish you the best for your career. And I'm pretty sure this will be excellent. And you will probably let us know a lot of new interesting stuff in the future. Voilà.
Thanks.
Congratulations. Bye.
Thank you.
Congratulations.
So is it my turn to say thanks? I don't know.
You can say something now. Yeah,
I will say some words then. Yeah. So yeah, I mean, this has been an amazing experience. It was always a bit of a gamble for me to start to go into biology. I didn't like biophysics in university. So I never expected I would end up doing biophysics. And I mean, I've really grown to love the complexity of the systems and just the science in general. This has been a really learning experience, both from the side of just like learning the material, but also from learning about myself and what I enjoy to do. So that's been amazing. And I mean, I have to thank you so much, Tom, for giving me this opportunity for. Maybe I'll tell the random story of how I ended up doing this thesis, because I had a course. I took one course of Tom's when I was studying in Antwerp, and I took one computer science course with Tom in Brussels in my final year, and it went well, and I really enjoyed the course. And at the end of the last day, I forgot my scarf, and I got an email the next day from Tom saying, "Hey, Nathaniel, I think you forgot your scarf. By the way, if you are interested in doing a thesis, let me know." And so that kind of kicked it all out. So sort of these random thing that happened that started it. But I'm really happy I forgot my scarf that day. And it's really been amazing to work in IB Square with all of the incredible colleagues I had there. I mean, the vibe has just been so awesome with everyone working there. It's so fun to go to work and work with all these people, even though like research wise we do different things. It's always been really fun. And then when halfway through my thesis, when I was feeling kind of a bit, I guess we had some setbacks with some of the projects and I didn't really know where to go. And then you suggested that we bring in Sophie as my co-promoter. And I was super happy on the one side, because I thought, I was feeling very nostalgic for my physics days in university. And so it was like, and Sophie is a physicist, so we can do like real hardcore physics stuff. And so I was really excited. And it went great. That's all I can say. Like it was fun to kind of discover this field together with you. And then you bringing your brother on for this project was also really, really nice. I think I learned so much doing that, and I really became excited as well. So like you said, I really hope we can do a bit more, continue this project a bit as well. So yeah, thanks to both of you. It's been really an incredible time. I'm going to miss the IB Square a lot, but I won't be too far. I'll be just up in London.
You're always welcome to come back.
Yeah. So yeah, I guess that's my thanks. Yeah, thank you.
Thank you very much, Nathaniel. And I guess Bartolo and Dominique, that's where we close.
Nothing to say on my side.
Yeah. The official part was already finished,
Yeah.
Okay, so maybe I would like to thank everybody in the room who came to the public defense.
Congratulations again.
Congratulations. And well, we can once again we want to thank the external jury members for their effort in assessing this thesis. Thank you from both universities.
Thank you very much.
Yeah.
And hopefully we can have a beer as soon as all this COVID stuff is behind us.
Yeah.
I keep
let's
you
hope.
to it, Tom.
There's going to be a lot of parties. I'm already been in so many juries in the meantime. So thanks a lot for all of you, and I hope to see you soon.
Yeah.
Bye.
Bye.
Bye. And thanks to Anne and Charlotte for all the assistance with technology stuff.
Take care.
You're welcome.
No
Bye-bye.
problem. Bye.
Bye.
I guess you can stop streaming, Charlotte.
Yeah.
Never.
I think
My fame.
Never.
My fame.
We still.
So actually, is this now recorded?
Nobody.
Is this now recorded on YouTube or not really?
Yes, it will be recorded on YouTube, indeed.
Okay, very good. Then we can keep this for posterity.
Yeah.
I like they have opinions on which periods. Someone said mustache period was the best.
No.
They're going to be